And now we're ready to kick off our first presentation of the day. Please join me in welcoming to the stage Judy Abrams. Judy is a member of OCRFA's Leadership Council and past president of the Ovarian Cancer National Alliance, and she's here to tell you a bit more about the Mello Abrams Lectureship. Judy.
Thank you, Audra. Good morning, everyone. Good morning. I hope you had a chance to explore the agenda and have been meeting some other fabulous attendees. My name is Judy Abrams, as Audra just said, and I am a past president of the Ovarian Cancer National Alliance. 14 years ago, I lost my best friend, Judy Mello, to ovarian cancer. To honor both Judy Mello and me, and our friendship, Jay Schwam, her husband, established the Mello Abrams Annual Scholarship Lectureship, which supports our ability to invite leading authorities in the field of ovarian cancer to address this conference. First diagnosed with ovarian cancer in 1994, after having been diagnosed with breast cancer in 1986, Judy Mello lived her life fully and courageously, without despair, and always looking forward, never back. She was also sustained by the love, affection, and support of her family and her many friends. But what allowed her to live her life as fully as she did during her illness was her incredibly positive attitude that she had until the very day she died. She lived each and every day guided by that spirit that she preached to one and all. Every day is a beautiful day. It's only the weather that changes. Now it's my great pleasure to introduce our esteemed speaker, Dr. Ursula Matulonis. Ursula Matulonis is Director and Chief of the Division of Gynecologic Oncology at the Dana-Farber Cancer Institute and Professor of Medicine at Harvard Medical School. She is the first recipient of the Brock Wilson Family Chair at the Dana-Farber Cancer Institute. She co-leads the ovarian cancer program within the Dana-Farber Harvard Cancer Center, and her research focuses on developing new targeted therapies for gynecologic malignancy with a specific interest in ovarian cancer. Dr. Matulonis serves on the National Comprehensive Cancer Network Recommendation and Guideline Committee for Ovarian Cancer, the Massachusetts Ovarian Cancer Task Force, the NRG Ovarian Committee, and the Scientific Advisory Board for the Ovarian Cancer Research Fund Alliance, among others. After receiving her medical degree from Albany Medical College, she completed an internship and residency at the University of Pittsburgh, followed by a medical oncology fellowship at the Dana-Farber Cancer Center Institute in Boston, Massachusetts. Dr. Matulonis will be able to take some of your questions after her presentation, so please write your questions on the note cards on your seat, and a volunteer will be around to collect them. Now, with great pleasure, please join me in welcoming Dr. Matulonis.
Thank you so much. Thank you. Thank you. Thank you so much. Thank you so much, Judy, for that lovely introduction. It's a pleasure to be here this morning. I love this organization and am very passionate about conducting research that helps women with ovarian cancer. And I think we're going to talk about a number of those different topics this morning. So it's a great pleasure. I don't know if those of you who are tennis fans, but there's a match on last night at Wimbledon between Nadal and Djokovic, which is still going on, but the women's final has been delayed. So I will check during our break, but this is 8 million times more important. So I'm going to really talk about new treatments for ovarian cancer and touch upon a number of different topics. And then hopefully there's going to be enough time for questions because I really want to get to them. So I have two agenda slides because there's so much to talk about. Really wanted to talk about kind of the foundation of ovarian cancer and from which a lot of our treatments have now emerged and how this has been so crucial to making treatment strides for ovarian cancer. So talk about the ovarian cancer pathologies and their differences. Secondly, the genetics of ovarian cancer and how the genetics are different within the different subtypes of epithelial ovarian cancer. And then briefly talk about some of the ongoing clinical trials for women with newly diagnosed ovarian cancer, one of which is about to report out at the European Society of Medical Oncology meeting just in a few weeks. And there was a press release, so I'll talk about some of those results. And then also talk about the following new therapies. So PARP inhibitors aren't so new anymore, but really to bring everyone up to date where all the FDA approvals are and where we stand with PARP inhibitors, current indications, new studies that have reporting out or are reporting out, maintenance trials, and then really focus on some of the combination strategies that are ongoing in many cancer centers around the United States and the world. Immunotherapy, where we stand with simply checkpoint blockade, drugs that you read about on television, and then some discussion around vaccines, also combination strategies with immunotherapy agents, antibody-drug conjugates, which are, I think, not had to date. There's no approval in ovarian cancer, but there likely will be one in 2019, and talk about ADCs, several in testing, combinations of drugs, and then some new drugs. And this is by no means an exhaustive list. It's really just a very early list, some of the trials that we're doing at Dana-Farber, plus elsewhere. And then also for future directions, really dependent upon your questions that you may have. So we know that ovarian cancer is now one cancer. However, trials in the past really viewed it that way. So therefore, the advances have been slow, but they're not so slow anymore, really because of this recognition that, yes, there's histology. So there are histologies of ovarian cancer that come back on pathology reports that experts in gynecologic oncology pathology are able to render through their eyes under the microscope, looking at the microscope, and also through special stains that they succumb that tissue to. But we also know that each of these subtypes have a different and unique and distinct molecular underpinnings. So for example, serous, high-grade serous or high-grade endometrioid tumor. It's the most common ovarian cancer, type of epithelial ovarian cancer. Up to 50% of all high-grade serous cancers have some underlying problem in the inability to repair DNA. Later on this afternoon, Dr. Alan DeAndrea will be speaking specifically around this, and he is an international expert on DNA repair, so you'll hear about that from the best. The cancers are also associated with P53 and BRCA mutations, and I'll talk a little bit more. There are other mutations that are important besides BRCA. So what are the clinical characteristics? Well, there's an increased sensitivity to drugs like carboplatin and cisplatin, but also an increased sensitivity to drugs like PARP inhibitors. Because these cancers have underlying DNA repair defects, and that's exactly how these drugs work. They pounce on cancer cells that have problems repairing their DNA. Low-grade endometrioid, a very unique, more rare tumor. They have specific mutations, so high-grade serous don't really have that many mutations. The mutations that we find tend to be things like BRCA1 and 2. But low-grade endometrioid have mutations in things called PTEN, ARID1A, and then mutations within the PI3 kinase signaling pathway. These are cancers that are potentially more sensitive to hormonal therapies. Still are probably using chemotherapy up front for certain individuals. These cancers tend to be lower grade, or lower stage and lower grade. Low-grade serous cancers, a really distinct entity. There's KRAS mutations present, BRAF mutations present, and hormonal therapies are particularly successful here. in addition to MEK inhibitors, these drugs called MEK inhibitors. When I mentioned these genetic characteristics, so these characteristics will be found mostly, and I'll talk about this later, on these tests called next generation sequencing. The top pieces, the mutations, are not going to be found necessarily in a pathology report, so that you need to do extra testing to the cancer to find out these different characteristics. Clear cell cancer also have abnormalities when PI3 kinase pathway, ARID1A, PTEN. These cancers can be there's a very heterogeneous bunch of cancers. Some can be highly sensitive to platinum-based chemotherapy. Some can have some intrinsic resistance to chemotherapy. So it's important on these cancers, again, to really do next-generation sequencing. I'm just going to put my phone down here. To really understand the genetics of this tumor. And then lastly, mucinous tumors. These cancers behave more like gastrointestinal tumors, like colorectal cancers. They tend to have KRAS mutations, and they are often chemotherapy insensitive, or if they're initially sensitive, then they become more insensitive. And there are really a lot of drug development programs ongoing that are targeting this mutation called the KRAS mutation. When you see RAS mutations within cancers, it just tells you that that cancer is going to be a little bit more insensitive to chemotherapy. So I've already mentioned the importance of genetics. So this is, and I have a slide on this. When you look at a tumor, there can be genetic mutations within that cancer. Now the question is that mutation coming from the cancer? In other words, did the cancer grow, or did it grow because of that mutation? Or was it an inherited mutation? So we know that several hereditary mutations are known to contribute to ovarian cancer, and the two most common are BRCA1 and 2. BRCA1 we know is associated with about a 40% lifetime risk of ovary cancer. BRCA2 a little bit less, 15%. Obviously there's breast cancer involved for BRCA2. It's not just breast, but there's also pancreas, melanoma, and others. But there have been 9 other genes that have been identified now as being high risk. So the first 4 on that list, MSH2, MLH1, PMS2, and MSH6, are genes associated with something called Lynch syndrome. And Lynch syndrome is a combination of ovarian cancer, endometrial cancer, and colorectal cancer. So sometimes we can pick the sort of the high-risk nature of that cancer based upon someone's family history, but a lot of times we're picking these mutations up just because we're sending all of our patients with ovarian cancer to the genetic counselors. And then there are a host of others, PALB2, BARD1, RAD51C, RAD51D, and BRIP1. So thus, we're sending the patient to a genetic counselor, or the physician is doing the genetic counseling. We're not just doing BRCA1 and 2 testing anymore. There's something called panel testing that really looks at all these now several genes that are involved in the hereditary nature, so that there can be recommendations made to family members who may also carry that gene. So up to 10 to 15% of all ovarian cancer, and that's really focusing on high-grade serous ovarian cancer, may have underlying BRCA1 mutations. And also, it's important that within that somatic or tumor, there's also another percentage where we find on next-generation sequencing of the tumor. So I'd say up to 20 to 22 or 3% of all high-grade serous cancers have an underlying BRCA1 or 2 mutation. And it's important to know that we do find germline mutations even within a patient whose family doesn't really have any history of breast or ovarian cancer. Hence, the recommendation, this is now an international recommendation, that all women with ovarian cancer should undergo testing regardless of type of ovarian cancer, her family history, or her age. And this is, I was alluding to this before, so this is what the difference between a germline mutation, that's on the left. So every cell in the body is blue, including the tumor, meaning every cell in the body has this underlying mutation, as opposed to the right, where it's somatic or tumor. So all the cells in the body don't have this mutation, so do not have an inherited BRCA1 mutation, but just the cancer does. And those we pick up through this entity called next-generation sequencing. So here we go, next-generation sequencing. It's a comprehensive genetic analysis of the tumor. And the testing typically gives us the following information. So it gives us information on mutations. So that would be important in high-grade serous cancers. You're finding that 6% or so of all high-grade serous cancers where the cancer only has the BRCA1 or 2 mutation. The rest of the body is good. Copy number changes. So these are changes within genes that they kind of replicate, or there are too much of them, or they something called delete. The gene is gone. And that is a real noteworthy aspect of high-grade serous ovarian cancer, where there are lots of these copy number changes, sometimes pages and pages. We just don't know what to do with that quite yet, but I think that that's an important, it's important to note that and to potentially use that and exploit that some way for therapy. Tumor mutational burden, thinking about immunotherapy, and then microsatellite status. That test goes along with this kind of Lynch feel. It determines whether or not that the cancer may have an underlying susceptibility to immunotherapy. Microsatellite status, typically in ovarian cancer, is not present. So it's not by itself going to show us that the cancer is necessarily susceptible to immunotherapy drugs. And the testing is available at specific institutions. So we have our own test. Memorial Sloan Kettering has their own test. A lot of cancer centers within the United States will offer this testing potentially as part of a research testing, so it's not a cost. There are commercial facilities where you can send your tumor to, and then there are also certain foundations that will do this test for free, like Clarity. Clarity is a wonderful organization. I'm on their board, and they will take tumor and run it through a next generation sequencing and help with decision-making around treatment. Okay, so what are some of the directions for newly diagnosed ovarian cancer in 2018? So use of PARP inhibitors is post-chemotherapy maintenance. Addition of immuno-oncology, so these drugs called checkpoint blockade or checkpoint inhibitors. You see them on TV, like Keytruda, nivolumab, et cetera. The use of both PARP inhibitors and immuno-oncology agents. And then fourthly, which I'm not going to talk about too much, is the use of intraperitoneal chemotherapy, HIPEC, so heated intrachemotherapy, after 3 cycles of chemotherapy when someone's newly diagnosed. Some centers are doing this, some are not. So these are some of the, and I'm not going to go over this too much because these trials are just starting. So these are studies for women who are newly diagnosed, for women who have undergone surgery and then are getting chemotherapy, or the surgeon makes the decision that they're going to receive 3 cycles of chemotherapy, then surgery, then more chemotherapy. And these trials listed, some of them are ongoing and some of them are planned. For example, the first study, the TSR-042 niraparib, is planned. We have a protocol in hand, but it hasn't started yet. And these trials are really asking the question, does adding an immuno-oncology drug or immunotherapy to upfront chemotherapy make a difference in terms of how women do? And then adding a PARP inhibitor as maintenance therapy, does that help? And so these are for your information, just to keep track of. And then the next slide is talking about maintenance. So this is somebody has surgery, they finish up chemotherapy, and now they're going to go on some type of maintenance therapy. In the United States, that maintenance therapy from an FDA standpoint could be bevacizumab, or it could be no maintenance therapy. The first trial on that list, called SOLO-1, and this is a study, this is how this study is designed. So this is for women whose cancers have an underlying BRCA mutation. So there's either an identified germline mutation through blood, or next generation sequencing looks at that tumor and said, aha, there's a bad BRCA2 mutation there. So those women finished up chemotherapy and then went on 2-to-1 randomization of either the PARP inhibitor olaparib or placebo, and were followed out. So this trial started a number of years ago, and the AstraZeneca just released a press release just literally a few days ago, showing that the study met its endpoint. In other words, the women receiving olaparib had a better cancer remission time than women who did not receive olaparib. So this will be applicable to a number of folks, and I think we're going to wait to see what the data is. The data is going to be presented at the European Society of Medical Oncology in October. So look for those results. I'm sure they'll be talked about in the press, as well. We'll be talking about it as well. All right, some of the new directions for recurrent ovarian cancer. So PARP inhibitors, yes. Combination strategies, certainly. There are lots of trials ongoing for combination strategies. Novel drugs such as antibody drug conjugates, and then immunotherapy strategies. So PARP inhibitors. And I'm doing a PARP inhibitor session later, which I'll get more into the nitty-gritty aspects and answer potentially some questions. But I'm going to first here talk about how do PARP inhibitors work? Well, they really work in two main mechanisms, although the enzyme PARP is a very important one and it has multiple activities, but around DNA repair. So when you're thinking about how does a PARP inhibitor work, it inhibits the enzyme PARP, and basically the enzyme has an ability to do is enzymatic work. So the PARP inhibitor prevents PARP from doing that. And PARP itself fixes little breaks in the DNA. So a break happens in the DNA, the PARP enzyme rushes, I describe it as an ambulance that rushes to the site of the break within the DNA. And if something gets in that way of the PARP enzyme working, then the break does not get repaired and the cell eventually ends up dying. So that's one way. And then secondly is called PARP trapping. So when the break happens in the DNA, the PARP enzyme, again, rushes like an ambulance, rushes to the break, and then kind of climbs on that break. Well, the PARP inhibitor causes that enzyme to basically get stuck. So it can't, because it has to go on and then flops off. But it gets on, it gets stuck, then the rest of the secondary responders, so the firemen, the policemen, can't do their job. So that's really how, and how I think about it, very simplistic way, how these drugs work. And the PARP trapping is actually a very important part of this. And these are the three medications. So I put the real name and then I put the commercial name next to it. So niraparib, olaparib is Lynparza, or niraparib is Zejula, olaparib, Lynparza, and rucaparib, Reca. And for those biochemists in the room, I also put the chemical structure down. So the diagram in the middle is taken from a review just published a few weeks ago, and it lists the different PARP inhibitors. So the ones that are approved for ovary are in the middle. So rucaparib, olaparib, and niraparib. Veliparib is on one end, and then talazoparib is on the other end. And that red line at the bottom shows you the ability of these drugs to trap PARP. So these are the three that are available in ovarian cancer. So even though there's it looks like niraparib is a better PARP trapper than rucaparib and olaparib. Rucaparib and olaparib are probably similar PARP trappers. Niraparib is probably maybe a little bit better. But from a clinical standpoint, to date, that hasn't made a difference in terms of the clinical trial results. So PARP inhibitors now have two indications from the FDA. One is this, and these are agents that have been identified as being particularly active in cancer cells that have underlying BRCA mutations, because we know that the protein BRCA, or BRCA1 and 2, are involved in repairing DNA. So if there's some underlying, again, problem with that, then those types of cancer cells are going to be sensitive to drugs like PARP inhibitors. So in all of these settings, so these are in a very mixed population, but this is what led to the FDA approval of olaparib, the first one. Secondly, rucaparib. Niraparib data just presented at our oncology meeting in Chicago a few weeks ago at ASCO, American Society of Clinical Oncology, with very comparable results. So I wouldn't be surprised if niraparib is eventually approved in this indication as well. So this is using the PARP inhibitor as treatment by itself, the oral PARP as treatment by itself for women whose cancers have underlying BRCA mutations. You can see the response rates range from 30 to 50%, and the durations of response, how long that cancer stays in remission for, is around 7 to 9 months. And these tend to be in more heavily pretreated situations, although rucaparib is a little bit less heavily pretreated. But I think that the niraparib data gives you promise that these drugs can work even in somebody who's received a number of different lines of chemotherapy. The second place where PARP inhibitors have been approved is in the maintenance strategy. And what that means is that somebody has a recurrence of ovarian cancer. It's deemed platinum sensitive, meaning there's more than 6 months between finishing last platinum and then evidence of the cancer coming back. Somebody receives carboplatin chemotherapy. It could be carboplatin gem, carboplatin doxil, carboplatin paclitaxel, enters into remission, and then goes on a PARP inhibitor. And there have been 3 trials, and I have a few slides of that have tested that strategy of somebody who completes their chemotherapy for recurrent disease and then goes on a PARP inhibitor, or actually went on their maintenance trial or placebo studies as well, that it helped answer that question. So this is PARP inhibitor maintenance, platinum chemotherapy, enter remission, go on PARP inhibitors. And then there's also Avastin or Bevacizumab maintenance. So the two have concomitantly developed simultaneously, but there's no trial to date, there will be, but no trial right now that asks the question, should we be using both of these drugs together? So for right now, we're using either one or the other. So these are the 3 PARP inhibitor maintenance trials. The NOVA study, which was the first, and that was the first PARP inhibitor approved for maintenance therapy. It's approved in 2017. It's also approved in Europe. SOLO2, the olaparib data, so that's olaparib. That trial was only done in cancers that had underlying BRCA mutations. NOVA was all types of ovarian cancer, and Ariel 3 was very much like the NOVA trial design. All 3 were phase 3 trials, randomized and placebo-controlled, and treatment was started after completion of chemotherapy. So it's important that because these agents have bone marrow suppressive capabilities, that you do not combine a PARP inhibitor with chemotherapy. The level of bone marrow suppression is too much. And there have been other studies that have looked at that, and you end up having to dose reduce the level of chemotherapy or dose reducing the level of PARP inhibitor. So right now they're used sequentially as opposed to combined. All 3 trials showed remarkably similar results, and all showed an improvement in how long the cancer stays in remission for. And those results were particularly striking for women whose cancers had BRCA mutations, so BRCA mutated cancers. There was another group that had something called homologous recombination deficiency testing, or HRD testing, where there was also a remarkable improvement in the length of how the cancer stayed in remission for the PARP inhibitor versus placebo. But then even in women whose cancers were underlying BRCA negative, or we call wild type, and had HRD negative testing, there was also an improvement in keeping the cancer in remission. So the FDA has now given approval for all 3 of these PARP inhibitors. They're now FDA approved in patients with recurrent ovarian cancers, maintenance following response to platinum-based chemotherapy, regardless of histology and regardless of the underlying BRCA mutation status. So the FDA gave a very broad approval to these cancers. So I think the questions that come up and are important to discuss with medical teams are PARP inhibitor versus bevacizumab maintenance. I mean, I think at some point there are going to be trials that are answering the question of looking at both together. But for right now, as I mentioned, we don't have that. And if the decision is a PARP inhibitor, how do you select that? Well, that's a whole nother question, which I will, if you guys come to the PARP inhibitor session, and I'm happy to answer it offline too. And the breakout sessions are all interesting, so I don't want you to necessarily come to mine. I'm like saying you have to come to mine to hear this, but there are other good ones too. So given the comparable efficacy of the 3 agents available for treatment, other characteristics will need to inform this, and I'll get into more of the weeds of this: the toxicities, the drug-drug interactions, the schedule, the price, and then special clinical situations. So 2 of these drugs actually enter into the central nervous system. One of them doesn't. So olaparib and niraparib actually seep into the central nervous system, but rucaparib does not. So I have a few women who have potentially carcinoma meningitis or brain metastasis. So if I want to get that drug into that spot, I'll use one of the 2 that I obviously get into the central nervous system. Okay. And then these are 2 other maintenance studies, as I alluded to, the bevacizumab or Avastin studies, Oceans and GOG 213. I'm not going to go into them too much, but maintenance scheduling time for Oceans and GOG 213, the Avastin was started with the chemotherapy, because Avastin doesn't have any significant bone marrow suppression, so it was started with the chemotherapy. So that decision about bevacizumab maintenance or PARP inhibitor maintenance has to be made at the time that the chemotherapy is being decided, and then the pathology of the cancer. So for the PARP inhibitor trials, the histology that was allowed into those studies was high-grade serous, but for Avastin trials, it was agnostic to histology. And I think you're going to mention the questions arise as to which is the best strategy, and it has to really be an individualized decision. Okay, this is a summary of the current PARP inhibitor FDA indications, and I just went over this, but basically olaparib, rucaparib, and niraparib all have maintenance indications regardless of histology, regardless of underlying BRCA status. And for now, olaparib and rucaparib have single agent treatment indications for women who have recurrent cancer and have an underlying BRCA mutation. Olaparib is technically just a germline mutation. Rucaparib is somatic and germline, but in the end, it shouldn't matter because the somatic mutation, if it's considered a deleterious mutation, functions just like a germline mutation. Okay, so, okay, let me go over these really quickly. How much time do I have left? How many? 30 minutes, okay. But I also want to leave time for questions. So what ends up, what ends up happening, somebody goes on a PARP inhibitor and the cancer starts to eventually grow, and that can be right away. I still have women on single agent PARP inhibitors for years now, so sometimes it doesn't happen. But if it does happen, there are lots of different reasons why that's happening, and I bet Dr. Deandra will cover this more extensively in his talk, but there are several reasons for this. The one on the top is the most common, and what ends up happening is that BRCA mutation that renders the cancer cell susceptible to a PARP inhibitor actually fixes itself. It fixes itself so that the mutation is no longer there, taking away that susceptibility. So that's called a somatic reversion or a restoration of the open reading frame. So that fixes itself. But these are just 3 examples of why a cancer cell can become resistant to a PARP inhibitor, but there are many, many others. The other 2 are more around sort of, again, making a functioning BRCA protein. So where it was deleted or not working, you've kind of restored it in some form. So in that case, what we've tried to do over the past few years now is to take, we call it HR proficient, meaning a cell that has intact DNA repair capabilities, and then, but make that cancer cell HR deficient. So homologous recombination is a fancy term for how DNA can repair itself when it has these things called double-stranded breaks. So 2 breaks happen in both strands of the DNA. So we postulated that adding another drug to a PARP inhibitor can either make somebody whose cancer is growing through a PARP inhibitor more susceptible, or de novo, a cancer cell that you don't think is going to really respond well to a PARP inhibitor, but make that cancer cell more PARP inhibitor sensitive. And so there have been, these are just examples of, on our team at Dana-Farber, of different strategies of how we've tried to take a PARP inhibitor and add something to it to try to make that PARP inhibitor work better. And specifically, and I know, and I bet Alan's going to talk about this more, but I'm going to talk about PARP inhibitors plus anti-vascular drugs, so a drug called cediranib, but there are also trials looking at the combination of olaparib plus Avastin, PARP inhibitors plus immunotherapy drugs, and then PARP inhibitors plus drugs called heat shock protein 90 inhibitors. And these are the different teams. I think it's really important that teams are formed to really help address, from clinicians and clinical trialists all the way to basic laboratories that are working on DNA repair, immunotherapy, et cetera. So for example, the Stand Up to Cancer Ovarian Cancer Dream Team, led by Alan and Liz Swisher at University of Washington, that's been a very productive collaborative effort across the United States, looking at PARP inhibitors plus PI3 kinase inhibitors, plus PARP inhibitors plus immunotherapy drugs, and I'll talk about that one later. So this is a trial that Joyce Liu, one of the faculty within our division, started when she was a fellow back in 2009. And we were just sitting around one day because at that point, Obama was president. He was president, and he had put out these stimulus grants to basically get teams together and so that we could hire more people. So we said, well, why don't we put together the PARP inhibitor olaparib? We know it works pretty well. And we added together another drug called cediranib, which is like an oral Avastin. So she did the phase 1 testing and then led to a phase 2 trial of either olaparib or the combination of olaparib plus cediranib in women with platinum-sensitive recurrent cancer, not as maintenance, but as actual treatment. And what she showed was that the combination significantly increased the time that the cancer stayed in remission for compared to olaparib alone. And additionally, when we looked retrospectively at some of the results, found that that combination really worked best in women whose cancers did not have an underlying BRCA mutation. So the PARP inhibitor worked very well in those women who had an underlying BRCA mutation, but if you did not, the combination was strikingly superior. So this has led now to 3 National Cancer Institute trials. The first is 9825, which is a phase 2 trial, which is completed. It was basically looking at the combination in both platinum-resistant and platinum-sensitive cancers and doing biopsies. And then the latter 2 were for platinum-sensitive, so carboplatin treatment versus the combination versus olaparib alone. And we just learned, the trial's been completed and accrued to, we don't have any results, so we'll look forward to that. And then the last was in platinum-resistant, and this was the trial just reported out yesterday. It's basically saying that the trial now can go on to the phase 3. So the phase 2 looks promising, so that GY005 will open. And that's basically the combination of olaparib cediranib versus non-platinum chemotherapy such as Doxil or topotecan. So, and this is a second effort led by Panos Konstantinopoulos in my group, looking at something called a heat shock protein 90 inhibitor. So we know that this agent, HSP90, is a protein. It kind of works, I explain it as a chaperone. So it's like a chauffeur. And it drives other proteins around the cell. So if you put a puncture in its tire or take its wheel away, it's not going to be able to drive around these important proteins, VIP, very important proteins around the cancer cell. So we know that very low doses of these drugs can help PARP inhibitors work better. So, and Panos, this is Panos in the right upper corner, several of you may know him, but he did some mouse work that showed that the combination is going to work better than either drug alone. So what's the key part here is the red line. So it's a graph showing over time, and if the line's going up, that means that the cancer is growing in the mouse. So the red line in most of these different experiments did better than either the single agent alone. So that led to another National Cancer Institute sponsored study of combining olaparib with the heat shock protein 90 inhibitor called AT13387. And that trial's ongoing. And he's now on, it's a dose escalation because we're really looking at safety. But so it's fullish dose of olaparib and then kind of layering on the HSP90 inhibitor. So we're not the only group that are doing PARP inhibitor combination work. This is a partial list of the different PARP inhibitor combinations that are going on right now worldwide. This is not immunotherapy, but these are adding small drugs, small molecules, or different types of agents. You'll notice none of them contain chemotherapy because of what I mentioned before, that adding chemotherapy to a PARP inhibitor, to a real PARP inhibitor, is problematic because of the overlying bone marrow suppression. A few words about antibody-drug conjugates. These are how antibody-drug conjugates work. So you've got an antibody, that's that big Y, and the open Y recognizes a protein on the cancer cell. So it's like a carrier pigeon. It's a pigeon that has its destination, and on the surface of it, the red lines are called linkers, and then the starish is a payload. So that is typically a very toxic, potent chemotherapy, but in very small amounts. So if you gave it like in an IV, we wouldn't do very well. But if you give small amounts and that amount is delivered to the cancer cell, that can be very potent. So the linkers, the red lines, can be cleavable or non-cleavable. They're all different sort of chemistry around these ADCs. And the payloads are of two types. One is a microtubule inhibitor. Basically, it attacks the skeleton of the cell. There's a cytoskeleton that keeps the cell round, and then DNA damaging agents. So this is another example of how that the blue one on the top is that antibody-drug conjugate, just what I showed. The orange circles are the payload of the drug. And what ends up happening is that the antibody-drug conjugate is internalized into the cancer cell, and then lysosomes basically cleave the drug, so the drug spills into the cancer cell, kills the cancer cell, and as the cancer cell is opening up, the drug can then hit nearby bystanders. So it's an interesting concept. It's not a new concept. These drugs are available in breast cancer as well as certain types of lymphoma. And these are the list of some of the agents that are being tested in ovarian cancer, and you can see the different targets. So there's a drug target called NaPi2b, the folate receptor alpha, MUC16 or CA125, which makes sense for ovarian cancer, and then a host of others. And then it's attached drug, and then where these trials are in ovarian cancer. So I'm going to mention a drug called Mirvetuximab, soravtansine, we call it Mirvetuximab, and the antibody-drug conjugate is against the folate receptor alpha. So I mentioned it binds to this receptor on the cancer cell. Approximately 75 to 80% of high-grade serous ovarian cancers have this protein on its outside surface. The drug that is the toxin, the payload, the orange circle or the star, is called DM4. And we initially in phase 1 testing saw very impressive response rates in women whose cancer had been heavily pretreated and platinum resistant. The most common adverse events were fatigue, some neuropathy. There's an eye toxicity that's not specific to this drug, but is specific to antibody-drug conjugates, and they're called corneal microcysts. So it's a blurriness of the vision, and it's important to keep the eye hydrated and moist, and it's a reversible toxicity. And there's a phase 3 trial that's now completed accrual called FORWARD-1, and that was comparing single-agent mirvetuximab to chemotherapy such as Doxil, topotecan, or Taxol. This drug just received FDA Fast Track designation on June 18th, and that means that the FDA will help facilitate its movement forward in terms of approval, obviously if the data warrants that, and we expect results from this trial in early 2019. Our group, as well as others, and this is again an international effort, looking at mirvetuximab combinations. And we've presented some of these results recently as just a few months ago. So adding Avastin to this antibody-drug conjugate, adding an immuno-oncology drug, and there's some nice laboratory data that would really give you indication that adding an immuno-oncology agent to an antibody-drug conjugate is a good thing and a bad thing for the cancer cell. And then adding carboplatin to mirvetuximab. Mirvetuximab is, what it doesn't have, I'd mentioned those side effects, but it does not have hair loss. So that's a very positive part. And this is some of the results that I presented at a meeting earlier this year. And these are for cancers that have that receptor positive. So you have to take the tumor that's stored in the lab and then stain it for the folate receptor alpha. And if it's positive, then that means somebody can go on and receive this drug. So we saw in tough cancers, very promising response rates and duration of response. Okay, immunotherapy. So I'm going to talk about checkpoint blockade, the drugs that you see on TV, like Keytruda and nivolumab, combinations, as well as some introductory comments about cancer vaccines. So this is data that I present, and I'm presenting this because I think it's a large trial. It's the largest study of single-agent immuno-oncology, because I get a lot of questions about, well, can I get Keytruda or can I get nivolumab? And there are a lot of patients enrolled in this. I just presented this a few weeks ago. So this was single-agent pembrolizumab. So pembrolizumab is something called a PD-1 inhibitor. So PD-1 inhibitors basically get in between this agreement between the cancer cell and the immune system that the immune system is not going to recognize it. So we call it a handshake. But these drugs called checkpoint blockade or checkpoint inhibitors get between that handshake and thus allow, theoretically, the immune system to attack the cancer cell. So there are two cohorts of women who are enrolled in this trial. This is an international study that had over 400 patients enrolled. So cohort A was women who had less heavily pretreated cancer, so 1 to 3 prior lines, but had to have treatment-free intervals between 3 and 12 months. The second cohort were more heavily pretreated patients, cohort B, and they could have any duration of treatment-free interval. So it could be 3 months or it could be 12 months, but they had to have between 4 and 6 prior treatments, and that could be chemotherapy mostly. So I'm putting this up here because we looked at the responses in 2 ways. We looked at the responses just across the board, looking at CAT scans or PET scan results. And the top line, the overall response rate, and I put a little, no, I know, it is, the overall response rate was only 8%. So this mirrors what other single-agent trials have shown, essentially very low response rates of 8%. So you'd like to see that. But then we also looked at the data based upon something called the CPS score. The CPS score is the combined positive score, and it looks at all of the cells within a cancer clump, and that can be cancer cells, that can be immune cells, that have PD-L1 associated with them. So it's all the PD-L1 positive cells, and the denominator are all the cells, so negative and positive. So the higher the CPS score, and you can see it goes from less than 1, 1 or higher, 10 or higher, that means there's increasing amounts of expression or presence of the PD-L1. So that's the binding partner to PD-1. So you can see that the higher the score, the response rate goes up. So 17% is actually quite promising. And we just got this data just before ASCO, and I think we're writing up the paper and trying to figure out how to move this forward. But I think these drugs could be potentially used for that subset. It's a small subset, though. Most ovarian cancers have lower CPS scores. Okay. And then these are some of the adverse events. Different than chemotherapy. A lot of gland problems, so hyperthyroid, hypothyroidism, but the other side effects are less. And some of the side effects that immunotherapy drugs can cause are colitis, so inflammation of the intestines, pneumonitis, inflammation of the lungs, hepatitis, inflammation of the liver. So all of these drugs can do that. You're getting ready to pull up 10. Okay, so Tapasio, I'm going to go through briefly. This is trying to figure out how to improve the activity of an immunotherapy drug. And in this case, this was done by adding a PARP inhibitor to it. And this is a huge collaborative effort with a lot of folks, the laboratory effort led by Dr. DeAndrea. So I'm just going to go through to the bottom line here. And this is the clinical activity. It's a complicated slide, but the bottom line is what's important. So the addition of pembrolizumab, this PD-1 inhibitor, to niraparib, oral PARP inhibitor, in cancers that were BRCA wild type, meaning BRCA negative, and HRD negative, led to response rates which were quite similar if we gave somebody a PARP inhibitor and they had an underlying BRCA mutation. So they were not overly super exciting results, but in a very specific population of women who have platinum-resistant cancer, but also have BRCA wild type, this may be a promising strategy. Okay. And then here are different immuno-oncology combinations that are ongoing, again, across the world, and we're actually now starting to do triplets. So 3 drugs together, and nivolumab, bevacizumab, and rucaparib is going to be run by my colleague Joyce Liu at our institution. It's not open yet. We're just adding rucaparib to the first 2, and then a trial called the OPAL trial, which is led by Tesaro, and Joyce is leading the first arm, TSR-042, which is their PD-1 inhibitor, plus bevacizumab and niraparib. Briefly about cancer vaccines. So what can a cancer vaccine do? And these are 3 situations. This is taken from a review from a colleague of mine that was just published a few weeks ago, Kathy Wu's group. So the first situation is the tumor on the left, those gray little boxes, doesn't really have any immune cells in them. So that we would think about as a cold tumor. But the vaccine might allow T cells, so the orange cells, to come on in and start the beginnings of an immune cell or an immune reaction. The second is if you already have some immune cells within the tumor, and it can amplify that. And the third situation is where you really got already a robust immune infiltrate happening within the cancer, and that can even make that, the vaccine can even make that more diverse. So as I mentioned, many ovarian cancers are immunologically cold, so that's why vaccines haven't gotten stronghold, and we've had to work a little bit more to try to figure out vaccination strategies. So this is also taken from Kathy Wu's review. In vaccine approaches, two important features are, one, how well the targeted proteins represent ovarian cancer. So what are you targeting when you're giving a vaccine? And then secondly, making it really personal, as opposed to just making a vaccine and giving it to everybody. So this is a review showing, so on the top is the tumor specificity of the antigen. So how well is that protein that you're targeting only present in ovarian cancer? And then on the left, going down, is how well are we personalizing it to a specific individual? So we know that University of Pennsylvania just this year reported vaccine results using autologous dendritic cells. So basically, these are immune cells taken out of a patient, and then they were exposed to ovarian cancer cells from the patient that had kind of been split apart. So that dendritic cell was able to see the proteins, they got reinfused, and hopefully then we're able to take those proteins and essentially hand them over to the T cell or present them to the T cell. And certainly, I think, very unique and very promising results. What we're starting to do is going over to the right and taking personal tumor neoantigen. That's another strategy that's ongoing, and I know the Penn folks are doing this as well, to generate a personal multipeptide neoantigen vaccine. So this is from my colleagues Patrick Ott and Kathy Wu, who first did this in melanoma. And I won't go over this, I'm sure you guys will get a copy of these slides, but basically taking tumor cells and then looking at different mechanisms of different like proteins and gene mutations, et cetera, and coming up with 20 proteins that are considered susceptible, that they're unique to the cancer, but that a T cell could also potentially recognize. And Panos is going to lead a trial that is not open. It's not open for a while, but just got submitted to IRB and scientific review committees. And the neoantigen satisfies the two ideal requirements for vaccine related to the specificity of the proteins to ovarian cancer and then personalization to that individual. The hurdles have been expenses to making the peptides. You think it'd be a simple thing, but it's several $1,000s per individual. So we've had to actually go to our development folks and fundraise for close to $500,000 to do this trial. Secondly, you need expertise in the determining which proteins are going to be considered these neoantigen proteins, yes or no. Our group, Kathy Wu, does this, and then manufacturing. It's a very hard thing to manufacture. So we're working through these three hurdles. Okay, in the last couple of minutes, just talking about other strategies and agents. So I mentioned anti-CD47. I think this is unique, and it's just starting. This is a protein that exists on a cell, and it kind of diverts the immune system away from, so it's called a do not eat me signal or do not attack me signal. And if you remove that signal, the immune system can potentially attack the cancer. And it was originally thought that maybe macrophages were the cells, but it may be other cells as well. So there are companies that are working on these. Prexasertib is another drug that's it's in clinical trials right now. It inhibits something called a cell cycle checkpoint kinase 1 and 2, and these are important proteins that detect evidence of DNA problems, of DNA repair. And in P53 positive cancer cells, just like ovarian cancer, these proteins are especially important. So by inhibiting those proteins, you might help the cancer cell die faster. So there's single agent trials, and then we're also looking at prexasertib plus other agents as well. Okay, and then I also mentioned this because I've talked a lot about high-grade serous histology, but there are also drugs that are being developed for other histology. So for example, endometrioid histology, regardless of grade. There's a drug called DKN-01 from Leap Therapeutics, and it is a monoclonal antibody against a protein called the Dickoff-1 protein that is especially important in endometrioid cancers where this pathway called the Wnt pathway is particularly active. It's a complicated pathway, but the bottom line is this drug is going to take away that protein and hopefully take away that driver for cancer growth, metastasis, et cetera. Okay, so concluding thoughts. Research on ovarian cancer has tremendously accelerated and continues to do so. I mean, there are trials being released, companies that are interested. It's vastly different even than just a year or two ago. Advancements have been made with histological and further molecular understanding of ovarian cancer. That's been critical. I think many novel clinical trial strategies are ongoing, and you've heard about them here, combination therapies, new drugs, et cetera. And I think PARP inhibitor development has been transformative. It's been transformative really because the drugs work. But secondly, it's the first time that the FDA had approved a drug since 2006, when carboplatin gemcitabine was approved, then 2014. So that led researchers, companies, everyone to say, wow, we can get drugs approved. So that has unleashed a lot of research efforts and individuals working on this cancer. And I mentioned teams are necessary to do these studies from clinicians, trialists, basic science labs, folks in between, and funding is absolutely 100% vital. And I know some of you guys are going to Capitol Hill on Monday, so just pound on them that we need the funding. I know, I agree. All right, so I'm happy to answer any questions that I saw a few cards passed around.
Okay, so we have lots of good questions. We only have a few minutes, but I will say, if your question doesn't get asked now, don't worry. All the questions that you guys sent up, I will save for the Ask the Expert session. So I'll put them in the box, so they'll get a second chance at being answered.
Yeah, and if you can shoot them to me, we can answer them.
So
No, I mean, if I don't get to them offline,
sure.
And then we can make sure people are getting their questions answered. Yeah.
Good point. So we have some questions that all center around the idea of BRCA testing and retesting. Mm-hmm. So if you were tested and were negative years ago, should you be tested again? If you were tested years ago and were positive?
Yes. Yes. It's a great question. We have to do that because a lot of times our women who have been diagnosed with BRCA or have been diagnosed with ovarian cancer are doing well. They don't necessarily come back to see us. But even if you've been BRCA tested, let's say, there's not a really good answer in terms of timeline, but certainly 5 years ago or more, and kind of remembering that women who are doing well but never been tested should absolutely be tested, really because of the implications for families. So yes.
Do you have thoughts on aromatase inhibitors as maintenance?
Yeah. So I alluded to this before in my talk, but the low-grade serous histology, so that's the cancer that really has, and low-grade endometrioid for that matter as well, a higher susceptibility to hormonal strategies. And the drug called an aromatase inhibitor is a drug that prevents the conversion of testosterone to estrogen. So it's an estrogen blocker. And low-grade serous, low-grade endometrioid are very highly hormonally sensitive or hormonally manipulation sensitive. So now in our NCCN guidelines, we've actually talked about, across the board, anyone who's newly diagnosed with a low-grade serous cancer, once you've done treatment, to go on maintenance aromatase. And it looks like aromatase inhibitors work a little bit better than drugs like tamoxifen.
What is happening with metformin studies?
Yeah. So metformin, has been more tested in endometrial cancer, really because of the kind of this metabolic syndrome of hypertension, diabetes, et cetera. So it hasn't particularly been tested well in ovary cancer. I don't prescribe it because it has side effects, and it can interfere with clinical trial results, et cetera. So I think if you're taking it because your glucose is a little bit elevated, then that's okay. I don't think we know for sure if it's going to have any activity or not. I don't know. It's a good question, though.
Can people have more than one genetic mutation?
Yes, you can. The question was, can you have more than one genetic mutation? Yes. It's rare, but you can. And I think, so let me back up on that answer. So if you're talking about mutations, somatic or tumor mutations, yes. The cancer can have multiple mutations within the cancer. In terms of inherited mutations, it's more rare, but think, you've got mutations coming from mom, mutations potentially coming from your dad's side. So it can happen when somebody can kind of co-inherit mutations. I have a patient who's probably got a BRCA1 mutation from one family member and a BRCA2 from mom's side and another one from her dad's side. So it can happen, but it's pretty rare.
Can next generation sequencing be done on stored tissue?
Say that again. What?
Can next gen sequencing be done on stored tissue?
Stored tissue, absolutely. Yes. So as long as the cancer's been embedded in paraffin, yes, it can be. And most pathology labs will keep tumors for multiple years.
Just in the interest of time, this will be our last question. Again, don't worry, all your questions will be passed on to Ask the Experts. If you've had immunotherapy, does that negate you from having PARP inhibitors if you reoccur?
No. They're really, I thought you were going to ask, if you're on immunotherapy, could you go on another immunotherapy trial? Some trials are allowing now previous immunotherapy strategies, but no, immunotherapy, it has a different mechanism of action than a PARP inhibitor, so that's not a problem, no. Okay, thank you so much. Thank you for being here. Thank you.